Hi, this is Dr. Gray, and in today's chapter 12, we're going to begin to take a look at our genetics section, and we're going to begin looking at Mendelian genetics. And historically, there were a lot of misconceptions about how traits were passed from parents to offspring. So there was a time when people believed that within the sperm was a fully formed little human being, and that was how that the new person got transferred. And there was also a thought that offspring are only alike because of who their parents are. Everything about the egg, that females controlled all the traits. There was also a misconception that males control all the traits. There was an idea about blending inheritance where we were the average between our two parents, and that's not usually or always the case. And that inheritance was because of acquired characteristics, meaning that if something happened to the parent throughout its life, then that trait could be passed on to offspring. So all of these are examples of things that have been proven not to be true. And here is an example of that idea from 1640, where there was a tiny little human being all packed into the head of a sperm. Now, thoughts began to change in the late 1800s when researchers like Gregor Mendel started to look in and actually do some scientific experiments in the idea of how traits are passed from parents to offspring. Now, Mendel, who was a priest, is now considered to be the father of genetics from his research that he did. So Mendel picked his experimental species to be the pea plant. And there are some ideas about, well, why the pea plant? Well, these are a number of those ideas. Firstly, that there are many simple traits. A trait is a physical representation of the actual genes. So what does it look like, whatever that gene codes for? Keeping in mind that Mendel had no idea about genes or even DNA at the time. But things like flower color, seed color, shape, etc. were all things that were very easy visual cues. There were many different kinds of traits, and many of these traits were what were called true breeding traits. True breeding traits, meaning you could have parents that have two copies of that gene. The male and the female there are male and female components in each flower, something we'll learn more about in the second semester of biology. But each flower produces both male and female gametes. Each pea was a new individual. Therefore, with a plant producing many, many peas, this meant there are many, many offspring. Also, you can breed pea plants and generate lots and lots of offspring very quickly because they have short generation times. Also, taking care of plants was relatively inexpensive and really easy to do. So if we take a look at how Mendel recorded his results, we looked that he had a specific notation system, and we use this to this day, where the letter P, a capital P, represented the parent generation or the parental generation. We then have F1, so when you breed two true breeding plants, you get the first filial generation. So this is a cross between parents that produces offspring, brothers and sisters. If you then cross two individuals from this generation, then those offspring become your F2s, your second filial generation. And then so forth and so on. You get the following generations. Now, when you have what's called a self-cross, meaning because remember plants have both sperm-producing and egg-producing components within the same flower, then you can cross a plant with itself, and that's called selfing. And also, as I mentioned before, there's a true breeding component. When parents have the same traits always and the offspring have the same traits always, we'll come to learn that this means they have two copies of the same trait. So here, if we take a look at a example cross that Mendel might have done, we see the P generation, the parents. So these are both true breeding, meaning for the color purple flowers, meaning that every time they cross them, so we cross two parents, we get offspring. And then if we cross any two offspring, we get F2s, and all of these down the line will have only the purple color. So that means they're true breeding. We know now that that means they have two copies of the purple color allele for flower color. Now we can see that if you looked and you crossed parents that had a different variety of a trait, then you can produce hybrids. So if you have a true breeding purple, meaning if you breed this to another purple, a true breeding purple, you'll always get purples. But if you have a true breeding white, which does the same thing for white, and you cross these two, you find out that you get all purples. No blending, so no whites, but also no like light purples. They're all the same color purple. But then if you take two of these from the F1 and you cross them to produce the F2, all of a sudden you see, okay, purple, purple, purple. The white shows up again. So something happened between the parent generation and the F2, which meant that that trait was somehow carried through but not visualized in the F1. So some of the conclusions from Mendel's work were that blending inheritance was not the explanation. So the idea that you're getting something that is a intermediate, like a dark purple and a white make a light purple, that didn't happen in this case, although there are instances where that can happen. That the inheritance appeared to be due to some kind of particles, and Mendel didn't have a name for these particles. We'll later find out that those are genes. Now, some versions of a trait can be masked by other versions of the trait, and we call this dominant. So in the case of the peas, the purple was dominant and was able to overshadow the white if a copy of that trait was present in an individual. And so then other versions of the trait are covered up or masked, so we called those recessive. The recessive trait, we only see that if we have two copies of that recessive trait. So when we looked at the F1s, we didn't see it because all those F1s had one copy of the dominant trait and one copy of the recessive trait, and therefore the recessive one was covered up in those F1s. But if we then moved on to crossing two F1s, we see that that recessive trait can reemerge. So the F2 ratios, so this is in this second generation, we find that the individuals are in a ratio of 3:1 dominant to recessive versions, and that's always the case, 3:1. So there were 3 purple plants and 1 white plant. And so that tells us that the F1 generation, those monohybrids in the middle, they must have still had a copy of that hereditary particle for white, as well as a hereditary particle for purple. And the ratios further suggested, so that 3 to 1, that those hereditary particles must somehow have been contained into the gametes that the flower made. So the flower made eggs and sperm. So somehow those particles must have been in there, and then when those came together and fertilized, then that's how the traits came back together again. So remember that Mendel had no idea what these particles were. So what, in fact, do these particles turn out to be? So that was in Mendel's time. Let's look, and you know, a lot of his work still holds true, his ideas, but let's look at some more modern terms. So the trait is still something that Mendel used and we use as well. It's the characteristic that we can see. Something like flower color, eye color, these kinds of things that we can visualize, that's the trait. The phenotype is the characteristic version that we actually see. So the trait is flower color, but what is the color? If it's purple, then that's the phenotype. If it's white, that's another phenotype. The same is true for eye color. Now, alleles, we know that this information is carried on a gene, which is a region of the DNA, but an allele is a version of the gene, or it's a version of what codes for those characteristics. So we can have a dominant allele, and that would be one that covers up another allele. So that would be, in the case of the pea plants, the flower color, a purple flower. The gene that codes for this trait will have an allele for purple, and we represent that with a capital letter P. The recessive allele, that's the one that gets covered up, is going to usually get designated with a lowercase letter, for example, a small p, and that represents the white flower. Now, don't be confused that these are both P's and think they both stand for purple. The idea is that the big P stands for purple, and so we have it capitalized. The small p stands for something different, so therefore that's the white one. And then we had phenotype up here, which was what you see. Genotype, think genes. This is the actual version of the particle, the genes that are carried. So, for example, we have 2 copies of every gene, one on the chromosome that we inherited from our father, one that we inherited from our mother, and this is the 2 different copies. One might be dominant, one might be recessive, or both might be dominant, or both might be recessive. So if we have 2 copies of the allele that are the same, either 2 dominants or 2 recessives, that's called a homozygote. A homozygote. So 2 big P's, meaning 2 purple colors, or 2 little p's, meaning 2 white colors. So homozygotes are always true breeding because they have 2 copies. Now a heterozygote, hetero means different, so this means that you have 1 allele for one version of the trait and 1 for the other version. So we designate that with a capital P and a little p. So these are non-true breeding. And our last modern useful term is the zygote. So when we take an egg and a sperm, and remember eggs and sperm only carry 1 set of chromosomes, not 2, but when we put them together, the egg and the sperm together, we now have 2 copies of every chromosome, hence 2 alleles for every trait. And the very first cell, when the sperm and egg come together, is a single cell, and it's called a zygote, but it's diploid. Okay, some more terms. Gene. So the gene is the actual DNA region that codes for a particular trait. So Mendel called these hereditary particles. The locus is the region on a chromosome. Usually it's a gene. A wild type allele is the most common in a population. So that's the one that occurs most often. And a mutant allele, these are much less common in a population. And they may have been formed, this allele may have been formed by some kind of mutation. Now, often we think of mutation as being a bad thing, but it doesn't necessarily mean bad. It just means different. So Mendel came up with a couple laws after he did all his research. And the first law was the law of segregation. So segregation means to separate. So keep in mind that there are particles that carry information. He knew this. And that each true parent carries 2 copies for each trait. And these particles, he surmised that these particles come apart when the gametes form, such that each parent passes only 1 particle. So this come apart is where the segregation comes in. So that each parent passes only 1 particle into each of their gametes. So for example, if you are a pea plant that is purple and you have 2 dominant copies of that purple, 2 big peas, then if your sperm cells would only have 1 copy of each pea, each capital P. And your eggs would only have 1 copy of each capital P. But if you were a heterozygous plant, you might have some sperm that have a capital P and some sperm that have a small p. And the same is true with the oocytes produced. Then those particles, those genes that we now know, are recombined randomly during fertilization. It all depends on which sperm fertilizes which egg to find out what the offspring are going to be. And so to test this hypothesis, he thought that, okay, so if you take 2 true breeding parents, and you then look at the first generation, the F1, and you're making monohybrids, then each of those should have 1 copy of the hereditary particle. And he came up with a way to test that. And so he did. So he took the 2 parental true breeding plants, only purples, only whites. He crossed them and he got this plant. Don't worry about this white one yet. He got this one. So he said, so all of them were purple. All of them were purple. Then he said, well, what if I take another true breeding white and mix that into here? What do I get? Then he looked at his F1, and when he did that, he saw, I have, if I look at 8 plants, I have 50% purples and 50% whites. So we have a ratio of 1:1 dominant to recessive in the offspring of the F2. So why do you suppose that's the case? Think about that. So here, in order to help us figure out how these traits are passed, we use a tool called the Punnett square. It kind of looks like a little game board with 4 spots. And we take a look at what the parent has for possible alleles. And here we have 1 parent with 2 big P's, 1 parent with 2 little p's. And we put them on either side of the Punnett square. So say this is 1 parent, and it can make only gametes that are P, big P. And this parent can only make gametes that are little p. So we put those 2 little p's on this side. And then we start matching them up, because they're all going to match up. So we take a big P and a little p, and we go a little p and a big P and a little p, and so forth. And we look, this is our F1. So if we look at our F1, they're 100% identical. But what color would they be? Think about that. They have one big P, one little p. What color would they be? Now, if you take any two from this group, and then we put the same, we divide up their alleles over here. So we take this one, big P, little p, take this one, big P, little p. Now when we start matching them up, big P goes here, right? And big P and little p, big P, little p, little p, little p. So we can end up, if we cross two of these siblings, we can end up with seeing that white come back. So he wanted to determine whether an organism that has a dominant trait is a homozygote or a heterozygote. Okay, so remember, homozygote has two copies of the allele, or a heterozygote, which it has, these are two of the same copies. This is two different copies of the allele. So here we know that this one that has green P's has two copies of the recessive allele, but we don't know about this one because it could have two big copies of the Y, or it could have a capital Y and a small y. We don't know. So by doing lots and lots of test crosses, we can find out what we might get eventually. So remember, each parent carries two alleles for each trait. So here we always see them on either side of the Punnett square. And those alleles are going to segregate equally and randomly into the gametes, and you don't know which gametes are actually going to come together. The gametes are randomly going to combine in alleles, recombine the alleles, and we're going to form zygotes during the fertilization process. So if we did this test cross, we would see that we would end up with all yellows. We would end up with all yellows if we did, and so therefore we would know that this extra allele must be another capital Y, because if it was a small y, then this Y and this Y would form a green P, and that's what we have down here. So if this was a small y, then we would find out when we did the crosses that we would have a 50% combination in this F2. So in our crosses, we can predict what might potentially be the ratio of subsequent generations, and we'll do some practice ones of that. Before we get to that, though, we have to talk about probability. Probability is the likelihood that something will happen. So chance events are described by probability. And we've got some dice here. So if you roll a dice, there's a 1 in 6 chance that you're going to end up with a certain number. So the probability is given as a fraction, like I just said, 1 in 6, or as the decimal equivalent over the range of 0 to 1. So if you have a probability of 0, this means the event will never happen. So there's a probability of 0 that I'm going to roll a 7 with 1 dice, because there's no 7 on that dice, no chance at all. A probability of 1 means the event will always happen, and it's not due to chance in the least. So if I had a dice that had just 1 dot on every side, then that would be a probability of 1 that I would roll a 1, because there's nothing that could be different. Anything that's chance ranges between those values. So again, 6-sided dice, it'll be a 1 in 6 chance that I'm going to roll a number 4. So, for example, the change of now if you have a coin that only has 2 different options, then the probability that you're going to roll a heads on the coin is 1/2, and you can represent 1/2 as 0.5 or 50% chance. Now, you can multiply your probabilities. So the overall chance for an occurrence of 2 or more tries that are independent of each other is a product. You multiply their individual probabilities. So if you want to see what's the chance of rolling exactly a 5 on a dice one time, that's a 1 in 6. The chance of rolling exactly a 4 on a a single dice is 1 in 6. So the overall chance of rolling exactly a 5 on the first die and then a 4 on the second would be 1/6 times 1/6, which is 1/36. So that's a much bigger probability. It's much harder to get that result. And if there's multiple ways or tries for an outcome to occur, then the overall probability is the sum of the individual events. So, for example, if you can win a game by rolling a 4 or a 5 with one dice, then the chance of rolling a 4 is 1/6, or the chance of rolling a 5 is 1/6. So if you add those together, you will have 1/6 plus 1/6, 2/6. So you would have a 1 in 3 chance of getting either a 4 or a 5, so a better chance of getting that. So that's just a little introduction to the probability. So now we can take a look more. The chance of rolling a 4, a 5, and a 4 in any order, the chance of a 5 first is 1/6 times the chance of a 4 to be second is 1/36. But if the order doesn't matter, we might also roll a 4 first, which is 1/6, and a 5 seconds. So overall, the chance is 1/36 plus 1/36, because you can do it in any order, 2/36 or 1/18. Again, that's better chance than each of these individually. Okay, so the next thing that we'd like to take a look at is that we can combine those probability rules and we can put them into a visual for how traits are passed through generations. And this is mostly used for humans and a lot of time used for human genetic diseases. So there is a scheme that you can determine a map of traits passing from generation to another generation. It's called a pedigree. And the symbols that we use, a square for a male, an open square, an open circle for a female, a darkened square for an affected male, meaning having some trait that we're following, usually a disease or a disorder, a darkened circle for an affected female, a diamond if the gender is unknown, and a line through if we have a deceased male or female of any type. So here is a disease called alkalonuria, and we take a look. So here we have, and they've used a color scheme here of blue for affected, tan for unaffected. So we have a male that has the disorder, a female who doesn't. So then they have their child, they have a single child, and that is a male because it's square, and we see that that male has the disorder. Now that male then partners with this female, and then they have 3 children. They have one female who's affected, one male who's not, and one female who is not affected. Then we look at a third generation and we can see it keeps going and going. So after many generations, we can look back and see the passage of these traits to find out maybe if we can figure out what the status of these sort of missing alleles are, what they might be. Now, so far we've looked at crosses which involve only a single trait, like pea color or flower color. But what if we had a cross where we're looking at two different traits? So here we're looking at, again, using the pea, but we're looking at seed shape, either round, so that's a round, or wrinkled, so that's a wrinkled one, and seed color, either yellow or green. So we have Y representing the color and R representing the shape. So here we have the P generation of homozygotes, the F1. These are the double heterozygotes. So they're no longer monohybrids, they're now dihybrids because we have two different traits. And then we're going to have the F2. We'll be able to look at the genotypes and the phenotype categories. So Y stands for the yellow color, or the color, and the R stands for whether they're round or whether they're wrinkled. So if we do the crosses, we see that we have a yellow pea in the first generation that has two copies of the capital Y, giving us yellow, and two copies of the R gene giving us round. So we have a yellow round, and these are true breeding. Then we have another pea plant with two copies of the recessive gene, so two little y's, so the little y stands for green, and two copies of the wrinkled allele, and those are two little r's. So when we cross these two, we find out that everybody in the next generation, the F1, these are dihybrids. So they're going to have one dominant and one recessive copy of each of the two traits. So it's going to be yellow, even though it's a heterozygote, and it's going to be round, even though it's a heterozygote. So it is round and yellow. We then take two of these guys and we cross them. And we find out, well, what are the possible gametes? Well, remember that when the gametes are formed, you only get one version. So let's just look for the moment at the coloring. So for this one, it could only be a capital Y or a small y. So if we look here in one gamete, say these are sperm, one gamete, we can make a capital Y or a small y, capital Y or a small y, representing the color. What are our options for the seed shape? So that would be a capital R, we can do that twice, and a small r. Those are the only options that you can have, a capital Y with a capital R, a small y with a capital R, and then the same is true with the small r's. You have one version with the capital Y, one version with the small y. Now remember that that's going to be exactly repeated because we're crossing two from this generation. So we're going to have the same on this side. You go through the same process of matching everybody up, and what you come out with is the phenotypic ratio of 9 round yellows, 3 round greens, 3 green rounds, 3 wrinkled yellows, and only 1 wrinkled green. Because remember that you have to have all recessives to get this guy. So anytime you have a dihybrid, two traits, and you're doing a Mendelian cross, it'll always be 9, 3, 3, 1 for the ratios in the offspring of the F2. So this led to Mendel's second law, the law of independent assortment, which says that when you have multiple traits, then those traits will separate into the eggs and sperm separately. Now, Mendel was able to see this in his work, and this was what resulted here, that the Y separated independently of the R. You could have a Y with a capital R, or you could have a Y with a small r. It wasn't the case that every capital R had to go with every capital Y. And that's what Mendel saw. And this says that one trait has no effect on the segregation and recombination of alleles for other traits. Now, this is true as long as those alleles are on different chromosomes. So in this particular case, when Mendel was creating this law, he didn't know that caveat, but we know now that that is as long as the alleles that code for those traits are on separate chromosomes. Now, everything we've looked at so far was dominant covers up recessive, but there's a couple other opportunities. So this is called incomplete dominance, incomplete dominance. And that's where you get a mixing of the two possibilities. And we're going to consider snapdragon flowers. So when we have snapdragons, a true red flower parent, RR, and a true breeding white snapdragon, when we cross those, we end up getting a pink, which is a heterozygote, capital R, little r, but instead of having the red cover up the white, we get a mix. So incomplete dominance means that you get a mixing of the trait and something in the middle. Now, if you cross those two pinks from the F1, you'll get an F2 that looks like this. One red, two pink, one white. So I urge you to make a Punnett square for incomplete dominance and see what you end up with. And remember, you only get pink when you have the heterozygote. Now, codominance is another difference from classical dominance covering up recessive. And this one is often seen, not often seen, but is always seen in blood typing, but that's a common example. So here, instead of a mixing, you get both phenotypes showing up. So when we have the blood types A, B, and O, O is really 0, meaning they don't have an allele at all. There's no allele. So if you're type O, that means you don't have the allele for a blood type. There are two possible alleles. One is A, one is B. And if you inherit one copy of A from, say, mom and one copy of B from dad, it's not the case that one covers up the other or you get something in the middle. You get both showing up and you become an AB blood type. So remember, this I here stands for the allele, and this is the version of the allele. So you can be a type A if you have two copies of A, one you got from mom, one you got from dad, or if from mom you got an A and from dad you got an O. Remember, O is not an allele, but that A will still give you type A blood type. So go through and again, practice some of these crosses. Now, multiple alleles for a single trait can sometimes result, if you have multiple alleles for a single trait, you can, meaning two different alleles that code for different versions, you get a sort of possible differences. So here, if you have an allele, this is, say, one chromosome. If you have an allele C, an allele little CCH, and little c, then you have different copies of those alleles, and you can mix those up to get a variety of colors. And you see that with the fur color on these animals. And so where capital C is dominant over this recessive version, but this recessive version can be dominant if you have the capital C, this should be a little c. If you have the little c, ch, that's dominant to the little ch, and the little ch is dominant to the little c. So in order to be a white or an albino rabbit, you need to have 2 copies of the recessive version. These should be little c's, not capital C's. Then I want to move on and talk about sex-linked traits. So here, this means that the allele that codes for a trait happens to be located on those sex chromosomes. So we have regular body chromosomes, and of our 23 pairs, 22 of them are regular body chromosomes, and 1 pair are called sex chromosomes. And for humans, we have an X or a Y. And there are alleles that are located mostly on the X, the Y not really so much. Very few genes are located on the Y. So X and Y are not the same chromosomes. Like chromosome 1, they are identical as far as which alleles are present. There might be a different version of the allele, but it's always, say, the allele for her hair color or eye color. So here we see an example of a sex-linked trait. In these flies, the gene for eye color is located on the X chromosome. So you can see from the top, there's this like sepia color. Then there's brown. Then there is a red, and then there is a sepia. There's a white color. Now the red color is the wild type, and it's dominant to the white color. Only the red and the white alleles are on the X chromosome. The other eye color alleles are on the autosomes, the other chromosomes of the fly. So if we take a look and we see we have a fly, a female fly who has a red eye, and a male fly, sorry, it's reversed. The male has a red eye, the female has the white eye. We take a look, and this is the X chromosome, so females always have 1 X. Sorry, this is a male. Males always have 1 X and 1 Y, and the female will have 2 X's. The eye color here for the red or the white is located on the X chromosome. So here we have a capital W, so that means red. But because there's no allele on this white one, then this fly has a red eye. Whereas this female has 2 little w's, those are little w's, and so that one is the recessive color or the white. If we do a cross and we look at the female, what can the female produce? Only gametes that have an X and a Y, a little w. And the male can produce either X gametes, sperm, that have a big W or Ys. So we start doing crosses. We pull this one down and this one down. We're going to end up with a female because it's 2 X's. We have a big W and a little w, so a red eye. Pull this one over and this one down, and we get the same makeup, red-eyed female. We pull this one down, which is the little w, and the Y, but there's nothing on the Y, so we have a white male, and the same is true here. So the question is, could you ever get a white-eyed female by crossing these 2 flies? So if we look at sex-linked disorders, we find that if we have the sex-linked disorder, which most likely is on the X chromosome, then we see that the X gets passed to all her sons. The mom will pass an X to her sons. She passes an X to each of her offspring, but the father will pass only a Y. So if mom passes this X-linked recessive allele that has the disorder, the son will automatically get it. If she passes the X that doesn't have the affected allele, then the son will not get it. But you notice that the females are both, the daughters are both unaffected, even though this one becomes a carrier. Now, there are some alleles that are lethal, meaning that if you get, depending on if it's a dominant or recessive allele, if you get that copy, then the organism will not survive. So this is a very interesting cat, which is called the Manx cat. And all Manx cats are heterozygotes for the M allele with the L, capital L, gene. So they have one copy of this. MM homozygotes are normal old house cats that have a tail. This guy doesn't have a tail. Big M with the L version, these are the heterozygotes, and they end up being Manx cats, no tails. If the unfortunate offspring receives two copies of the lethal version of this combination, then those individuals will not survive. So if you cross two Manx cats, you'll produce an offspring ratio of 2 Manx to 1 normal. So do a cross where you have that they both have this makeup. They both have this makeup. Ooh, both have this makeup. And find out why the living cats will always be 1 normal and 2 Manx. Now, here's a tribrid cross, and we're not going to get into this in all kinds of detail, but remember those percentages and the probabilities that if you have 3 traits, like seed color, seed shape, whether the plants are tall or dwarf, you can start adding those up and find out how many in the F3, this would be the F3, we would see, this would be the F2. These are parents, F1s, F2s. We would have 3, 3, 3 that would be yellow, round, and tall. And if you multiply these, because the probabilities multiply, we would have 27 of these. So the most would be yellow, round, and tall. And you can go through and figure out what the percentages would be for each of those in a trihybrid cross. Now, there's a process called crossing over. We talked about this in meiosis, where bits of the chromosomes can swap during meiosis. We end up with different versions of the alleles. So the C's, that's the region of the allele, and these two have swapped. So now we have a version of this chromosome that has a small or a recessive copy of this allele. And we have a version of this chromosome that has the dominant copy. And these are considered recombinant chromosomes because they have different makeup. And so the C gene was swapped around. Now, our last bit, and I apologize for the length of this video, epistasis, saying that genes are not always completely independent, like Mendel thought, but rather they can interact to produce different phenotypes. So just kind of get used to the idea that they can interact. So here we have, and I'll put up some here, the mottled coat color of A is dominant to a solid coloration, such as gray or black. So in order to get black, you have to have the recessive gene. If you have a capital A, you're going to get this sort of tannish mottled color. But there's a separate gene at a different location, which is the C gene. And this one allows for pigment production. So this one is for whether it's solid or a mottled color, a mixing. But this one allows for whether the pigment gets actually formed at all. And if it will get formed if you have a capital C, the dominant, but if you have the recessive, then that organism, that animal will not produce any pigment. So if you look at the cross here, and this is a dihybrid cross, you will have some that would technically be mottled or black. This one would be black, but because they have 2 copies of the recessive C gene, they don't make pigment at all. So you end up getting an albino mouse that has no coloration, even though it was destined to be mottled or to be black. So just know that these genes don't act in isolation, that they may in fact be impacted by another gene. And so this goes on to explain that more, just what I had said. And so there's multiple gene interactions that can make a modified F2 ratio. So here it's the ratio is not exactly what Mendel had thought it would be. All right, thanks very much.